Beneath Antarctica's jagged ice, in waters colder than 0 degrees Celsius, life persists in a way that seems almost impossible. Fish swim freely in an environment where even steel can become brittle, and any ordinary animal would freeze solid. These fish have evolved remarkable adaptations that allow them to survive and thrive in these icy depths, defying the freezing temperatures that would normally be fatal. The Southern Ocean, surrounding the Antarctic continent, has water temperatures that hover around 1.9 degrees Celsius. At such temperatures, any ice crystal that forms within a living organism would pierce cells, destroy tissues, and end life almost instantly. Yet, Antarctic fish, particularly the Nototheniod family, remain perfectly functional, swimming, feeding, and reproducing in these near-freezing waters. The mystery of how they survive first captured scientific attention in the 1960s and 70s. Researchers studying Antarctic cod and related species noticed something extraordinary. When they cooled samples of the fish's blood serum below the expected freezing point, the serum remained liquid. It didn't freeze. This observation revealed that these fish had developed a unique biological mechanism that allowed their blood and body fluids to resist the formation of ice. The solution lay in specialized molecules known as antifreeze glycoproteins, or AFGPs. Antifreeze glycoproteins are extraordinary molecules, finely tuned by millions of years of evolution. Structurally, each AFGP consists of repeating tripeptide units, typically alanine-alanine-threonine, where the threonine residue carries a sugar molecule, specifically a disaccharide. These molecules circulate in the fish's blood and are produced as larger polyprotein precursors that are enzymatically cut into active peptides. This allows fish to maintain high concentrations of AFGPs in their blood, often in the range of 5 to 35 milligrams per milliliter, which is crucial for their protective effect. By producing multiple isoforms with varying lengths, fish create a dynamic antifreeze system that remains effective across seasonal changes and environmental variations. The mechanism by which AFGPs protect fish from freezing is known as adsorption inhibition. As microscopic ice crystals begin to form inside the body, AFGPs bind selectively to specific planes on the surface of the ice. This binding blocks further growth of the ice crystal. The ice that does form between the proteins is forced to adopt a curved shape, and this curvature raises the local melting temperature through a phenomenon known as the Gibbs-Thompson effect. This creates a small but significant difference between the melting point and the freezing point of the fish's body fluids, a phenomenon called thermal hysteresis. For Antarctic fish, this thermal hysteresis typically depresses the freezing point by up to 1 to 1.5 degrees Celsius, which is sufficient to keep them alive in waters that would otherwise be lethal. Some Antarctic fish have evolved even more extreme adaptations. Ice fish, belonging to the family Channicthiidae, have entirely lost hemoglobin and red blood cells, leaving their blood nearly transparent. While this would be fatal in most species, the cold Antarctic waters hold more dissolved oxygen due to the physics of gas solubility at low temperatures. Combined with a slower metabolism and larger hearts, ice fish can transport sufficient oxygen directly in their plasma to sustain life. Their circulatory system is adapted to maximize oxygen delivery despite the absence of hemoglobin, with greater blood volume, lower viscosity, and enhanced capillary networks. These adaptations allow ice fish to survive in a niche environment where red-blooded competitors would dominate under warmer conditions but fail in freezing water. The evolutionary history of these adaptations is equally remarkable. The genes encoding AFGPs arose through duplication and expansion of a trypsinogen-like gene. The repetitive tripeptide sequence is amplified to produce the polyprotein precursors that are then processed into functional antifreeze proteins. This genetic innovation is a stunning example of evolution shaping molecular structures to solve a physical problem. Interestingly, similar antifreeze proteins evolved independently in northern cod. A striking case of convergent evolution driven by the same extreme cold pressures in distant oceans. While these adaptations are highly effective, they come with trade-offs. AFGPs provide protection only within a narrow temperature range. As Antarctic waters warm due to climate change, these specialized fish may face increased competition from temperate species and potential disruption of their antifreeze systems. Despite these vulnerabilities, the biological solutions evolved by Antarctic fish have inspired human innovation. Antifreeze proteins are being investigated for medical applications such as cryopreserving organs, protecting frozen food, and engineering frost-resistant crops, demonstrating the profound potential of understanding natural adaptations at a molecular level. Antarctic fish exemplify nature's ingenuity in overcoming extreme environmental challenges. Through a combination of molecular innovation, physiological rewiring, and evolutionary specialization, these fish have transformed one of Earth's harshest environments into a viable habitat. Antifreeze glycoproteins, the physics of ice inhibition, and the radical adaptation of hemoglobin-free blood highlight the intricate interplay between biology and physics. In their silent gliding presence beneath the ice, Antarctic fish reveal not only the resilience of life, but also the ingenious solutions evolution can produce when faced with the seemingly impossible.